Hi, my name is Sarah Rogstad, and I'm a graduate student currently at the University of Pittsburgh, and I really like to study proteins. So by study proteins, I mean that I like to identify and measure proteins within a particular sample. So if we look at a sample from a healthy individual and compare it to a sample from a sick individual, and we look at all the proteins within there, and we compare those proteins, we can see better at a molecular level what's actually going on with the particular disease. So this field of studying proteins is called proteomics, and it's called that because it's similar to the study of genomics, which is looking at all the genes in your body, whereas proteomics is looking at all the proteins in your body. So the main tool we use to analyze this is called a mass spectrometer. And a mass spectrometer is basically just this big box that can measure small molecules. So particularly, I focus on a neuronal protein, so a protein that's in your brain, called dopamine transporter. So dopamine transporter, or DAT, is involved in a variety of diseases and disorders, including Parkinson's, ADHD, and schizophrenia, and it's also involved with drugs of abuse, such as amphetamine and cocaine. So what I do is I look at dopamine transporter and all of the other proteins that are associated with dopamine transporter and see how they're affected when you treat them with amphetamine. So we look in mice at both normal mice and mice treated with amphetamines, and we look at how the proteins within them change and what proteins are associated with dopamine transporter before the treatment and after the treatment. And so by looking at this, we hope to build a better pathway and better understand what's going on in your brain at a molecular level when you treat your brain with drugs like amphetamines. So by looking at the protein changes, we can understand better what's going on at a molecular level.